What if the very medicine meant to help you could instead cause harm or be completely ineffective just because of your unique biology? Here's where it gets really interesting and quite personal. Today, we're taking a deep dive into something called Cytochrome P450 enzymes, or CYP enzymes for short. Think of them as your body's incredibly precise personal detox factory, tiny but mighty workers, absolutely crucial for how your body processes drugs.
That's right. Our mission for you today is really to pull back the curtain on these enzymes. We'll explore exactly how they function, how genetic variations in your DNA can influence their activity, and even some surprising factors like inflammation, your diet, even your body's internal clock. All of these can profoundly affect how drugs work for you.
Yeah, our goal is basically to give you a shortcut to being genuinely well-informed. We want to provide those aha moments that help you truly understand why drug interactions happen and pave the way for a future of more personalized medicine, all without, feeling overwhelmed by complex scientific jargon.
Okay, so at their simplest, think of CYP enzymes as a big family of tiny, powerful processing units. They're mainly found in your liver and your gut. And they're built with this special component, almost like a chemical key, that lets them break down or metabolize a huge number of things. And that's not just medications, right? It's also compounds naturally in your body, like lipids and steroids.
You mentioned their role is truly critical. Could you walk us through what phase I drug metabolism actually involves? Is it sort of like a sorting hat for chemicals?
Ha! That's a fun way to put it. Yeah, in phase I, these enzymes are essentially altering the chemical structure of drugs.
Yeah.
Think of it as the first major processing step. Sometimes they might even take an inactive prodrug and sort of flip the switch to turn it into its working form. Then after this initial transformation, the drug moves to phase 2. That's where it gets tagged to become water-soluble, making it easy for your body to get rid of it. It's truly a complex assembly line inside you.
So if they're doing that much heavy lifting, are we talking about the majority of our daily medications being processed this way?
Precisely. Yeah, to put their broad impact into perspective. Nearly 80% of the oxidative metabolism of clinical drugs involves these CYP enzymes. They are absolutely foundational to how most medications work in your system. Wow, 80%. Okay. And within this vast family, are there a few star players, the real heavy hitters that process most of our common medications? Absolutely. Yeah, the main families we focus on in drug metabolism are CYP1, CYP2, and CYP3. And among those, some specific enzymes, or isoforms, are exceptionally predominant. So when we talk about the most predominant CYP isoforms, CYP3A4 immediately comes to mind. It's thought to handle over 50%, half of all CYP-mediated drug oxidation reactions. You find it heavily concentrated in your liver, and especially your intestine. That's where it plays a big role in what's called the first pass effect, which means basically it starts breaking down drugs even before they get fully absorbed into your bloodstream, so it can potentially reduce how much actually reaches its target. It's also found in your kidneys. And what's particularly significant here is that CYP3A4 is involved in the greatest number of drug interactions.
Right. So you might naturally assume, okay, if two drugs need the same enzyme, they're always going to clash and cause problems. But is that always the case with CYP3A4, even though it handles so many medications? Is there like a subtle insight here?
That's a crucial point. It's not always a straightforward competition.
Yeah.
Think of CYP3A4 not as a single-lane highway, but maybe a multi-lane intersection, or maybe a large warehouse where things can be processed at the same time. Drugs can actually bind to different separate regions of the enzyme's active site, or even occupy the site together, allowing both to be metabolized simultaneously. It's far more nuanced than simple competition.
That's fascinating. So it's not simply first come, first served. Okay. Are there other important members of this 3A family then?
There are. There's also CYP3A5. It has similar specificity to 3A4, but interestingly, isn't present in everyone. This means individuals who express both CYP3A4 and CYP3A5 have the potential for increased metabolism of certain drugs. And then there's CYP3A7, which is primarily expressed during fetal development and rapidly disappears after birth. Shifting to other key players, CYP2E6 is the next most important isoform. It metabolizes about 25% of all drug oxidation reactions, covering a wide range of medications like many antidepressants, antipsychotics, and important opioid analgesics.
Okay, so we've spotlighted the real heavy hitters like CYP3A4 and CYP2D6, but it's clearly a much larger team working behind the scenes, isn't it?
Indeed, the drug metabolism team is quite extensive. We also have CYP2C8 and CYP2C9. Together, they handle about 16% of these reactions. And these are particularly significant because they metabolize drugs with narrow therapeutic margins, things like phenytoin and warfarin, where even small changes in concentration can have really big effects. Then there's CYP2C19, responsible for metabolizing over 12 frequently used drugs, including common medications like omeprazole and lansoprazole.
So we've talked about these incredible enzymes, but here's where the U factor really comes in. It's all about polymorphism. If our genes are dictating this, does it mean we're all essentially labeled into different processing speeds for drugs?
You could sort of say that. Yeah, we categorize individuals into metabolizer phenotypes, things like poor, intermediate, extensive, which is the most common speed, or even ultra-rapid metabolizers. And these are dictated by the, over 2,000 described genetic variations. These single nucleotide polymorphisms, SNPs for shorten your CYP enzymes, these tiny genetic differences can significantly impact how active your enzymes are. This is truly the core of personalized medicine.
That's powerful. Understanding these genetic mutations holds immense potential then. It sounds like knowing them could help doctors predict how a patient will respond to a drug, and maybe even flag potential adverse side effects before they happen.
Exactly. And we're still learning. The 1000 Genomes Project, for instance, identified many non-synonymous coding SNPs variations that change the protein with over 1% frequency that hadn't been cataloged before. This just shows there's still so much to uncover about how our unique genetic makeup impacts drug metabolism and how we can refine that personalized approach.
Okay, so we've established how our unique genetic makeup turns us all into different kinds of metabolizers. But what happens when you introduce another medication into this intricate biological system? This sounds like where things can get complicated, where the dance of chemicals truly begins, right?
It absolutely does. And this brings us squarely to the world of drug interactions, or DDIs, specifically through the lens of these very CYP enzymes. When we talk about DDIs and CYPs, there are two main categories to understand: inhibitors and inducers. Inhibitors are substances that decrease CYP activity. They slow the enzymes down. This can lead to higher drug exposure in your body, potentially causing toxicity. For instance, a strong inhibitor can increase a drug's exposure by 5-fold or even more.
So potentially too much of a good thing, which could then be harmful.
Precisely. Then on the flip side, you have inducers. These are substances that increase CYP activity. They speed the enzymes up. This leads to lower drug exposure and can unfortunately result in the drug losing its therapeutic effect entirely. A strong inducer, for example, can decrease drug exposure by over 80%.
Wow. So it's not just about side effects, but potentially a drug you're relying on simply stops working. That's quite a revelation for someone who might just be thinking their medicine isn't strong enough.
It's a critical point to understand, absolutely. And, it's not always so straightforward. A strong inhibitor or inducer in general might still have only a moderate or weak effect on a specific drug, depending on how that particular drug is metabolized. It's definitely not a one-size-fits-all rule, which makes it a complex puzzle for healthcare professionals.
Right, context is key. And it's not just pharmaceuticals we need to consider, is it? Are there common non-drug substances that can also act as CYP inhibitors or inducers? Things we might encounter every day.
Absolutely. This is where it gets really surprising for many people. Popular dietary supplements like St. John's wort, curcumin, and diosmin can do this. Even lifestyle factors like tobacco smoking and everyday foods like grapefruit juice can play a significant role.
Grapefruit juice, yeah, I've heard about that one. And herbal remedies, in particular, they present a unique challenge, don't they? They're complex, and often variable composition makes it incredibly hard to predict their interactions with drugs. But we do have clear examples, right?
We certainly do. St. John's wort, for instance, is a very well-known inducer for multiple CYPs, including CYP2B6, CYP2C9, CYP2C19, and CYP3A4. It works primarily through its interaction with a key regulator called the PXR receptor. This is why if you're taking certain medications like, say, oral contraceptives or some antidepressants, St. John's wort can seriously reduce their effectiveness.
Okay, so that's drug interactions and even food and supplement interactions. But you mentioned inflammation earlier. Does that mean if someone's body is already fighting something, say chronic inflammation, it could impact how their medication works? This sounds like a whole other layer of complexity beyond just genetics or other drugs.
Precisely. Your body's overall state and environment play a profound role. It's not just about genes and pills. The body's inflammatory signals, those pro-inflammatory cytokines, you often hear about things like TNF-alpha, IL-1 beta, IL-6. These can actually tell the liver's CYP enzymes to slow down. So a state of ongoing inflammation might unknowingly reduce how quickly you process drugs.
So to give you some specific examples then, if your body is dealing with, say, a severe infection or chronic inflammatory condition, certain signaling molecules like TNF-alpha or IL-1 beta can reduce the activity of key enzymes like CYP2C8, CYP1A1, and CYP2C19. And quite interestingly, you mentioned IL-1 beta can even increase CYP3A5. That seems counterintuitive. But the bottom line is, what does this all mean for you, the listener? It means that if your body is in an inflammatory state, your drug clearance might be reduced, and that could potentially lead to adverse reactions because the drugs aren't being processed as quickly as expected.
It's an incredibly intricate regulatory dance. You have these nuclear receptors like PXR, CAR, and AHR, along with other pathways like NRF2, which responds to oxidative stress. These act as key xenobiotic sensors, basically detectors for foreign substances that regulate CYP enzymes. Even your circadian rhythm, your body's internal clock, can affect CYP activity. And that clock can also be disrupted by inflammation, further adding to the complexity. It's all interconnected.
Understanding these complex interactions is therefore vital for real-world applications. It's why advanced research models like hepatocyte co-cultures that mimic the liver's environment are so important. This knowledge can help healthcare providers adjust drug timing and dosage to avoid adverse reactions and achieve truly individualized treatment. For example, some studies show natural products like silibin from milk thistle can inhibit liver inflammation and restore CYP3A activity. And even certain monoclonal antibodies, sophisticated drugs, can restore the liver's detoxification capacity under inflammatory conditions.
Okay, now let's shift our focus slightly to an especially vulnerable population, children. Despite the clinical implementation of genotype-guided therapy in adults, using genetic tests to guide drug choice, these associations are much less established for pediatric patients. It's a significant knowledge gap.
So a study found that out of 48 drugs CYP pairs with high evidence in adults, only 10 were commonly used in children. And for the majority of those 10 drugs, robust pediatric data supporting genotype-guided prescribing is still lacking or, well, conflicting. This isn't just theoretical then. These genetic differences in CYP enzymes have had real, measurable, even tragic impacts that have forced regulatory bodies like the FDA to step in.
Absolutely. The codeine case is a stark and unfortunately very clear example of pharmacogenomics directly impacting patient safety in kids. See, codeine is a prodrug. It's inactive until your body's CYP2D6 enzyme metabolizes it into active morphine. That's what provides the pain relief. Now, if a child is a poor metabolizer due to their CYP2D6 genes, they won't get any pain relief because the codeine isn't converted effectively. But, and this is critical, if they're an ultra-rapid metabolizer, they can face severe toxicity, including respiratory depression and tragically even death because too much active morphine is produced way too quickly. There is ample pediatric evidence, including documented case reports, confirming this critical risk.
And the FDA responded directly to this data, didn't they? They issued strong warnings and contraindications against codeine use in children under 12, and specific warnings for adolescents and even breastfeeding mothers, because the morphine can pass through breast milk. This is such a clear case where pharmacogenomics can and does effectively guide therapy in children, directly impacting patient safety. It's a powerful, if sobering, example of what's possible.
It is. However, when you look at other drugs commonly used in children like ondansetron for nausea or risperidone for behavioral issues, while adult data on CYP interactions might exist, the pediatric evidence is often sparse or mixed or simply insufficient to broadly support widespread genotype-guided therapy yet.
Yeah, it sounds like the challenges in pediatric research are pretty significant, often a small number of studies, generally small participant sizes, and varied research methods. All that makes it really difficult to draw strong conclusions, which really underscores the ongoing critical need for more rigorous, high-quality research in this area to ensure all our most vulnerable patients are protected. So after all that, what's the key takeaway here? We've unpacked how your body's CYP enzymes are truly at the heart of how you respond to medications. This deep dive has shown that it's really not just about the pill itself, but about your unique genetic blueprint, other medications you might be taking, what you eat, your overall health status, and even the time of day. It's complex.
Exactly. Understanding this intricate biochemical dance is your shortcut, really, to being truly well-informed about your health and medicine. It also highlights why interprofessional collaboration among healthcare providers, doctors talking to pharmacists, talking to nurses, is so incredibly important for ensuring the best patient outcomes. Everyone brings a piece of the puzzle.
So the next time you take a medication, consider not just the pill itself, but that intricate biochemical dance happening within you, and how much more there is still to learn about optimizing that dance for your unique biology.